Advances in Translational Cancer Research and Therapeutics. And on this occasion, we are invited two eminent speakers. One speaker is from USA. She is Dr. Seemana Bhattacharya. And other speaker is Dr. Gopinath, and working as a principal scientist in CFTRI MYSORE. So on this occasion, I would like to cordially invite the Honorable Vice Chancellor, sir, on this occasion to deliver an inaugural address. Thank you, sir. Over to you, sir. VC, sir. Sir, over to you, sir.
Charya, can I start?
Yes, sir. You can start, sir. You can start, sir.
Okay. So, on behalf of the KRISHNA University, Machilipatnam, at the outset, it is my pleasure to invite today's the top-notch speakers, Dr. Seemana Bhattacharya and Dr. Gopinath, the great scientists, the academicians, the young researchers, and students fraternity for today's the one-day international the webinar on advance in the translation cancer research and therapeutics. I am sure that today's webinar shall explore the new ideas and concepts. Okay, one minute. Explore the new ideas and concepts on the topic of the cancer biology and the genetic cancer, biomarkers, the cancer, the prevention and the research, cancer, immunology, immunotherapeutics. I sincerely hope that this webinar will deliberate and discuss all the different facts of the existing topic on cancer biology and come up with the recommendations that will lead to a better, the healthier and merrier world. I must congratulate the organizing secretary, Dr. Brahmachari, and the faculty of the KRISHNA University from the Department of the Bioscience and the Biotechnology for their untiring efforts to organize this webinar, particularly in the selecting the top-notch invited speakers and looking forward to anticipate the many more webinars of such kind in near future. I wish all the best to all the participants of this webinar. Thank you. Thank you very much. Thank you, Chari.
WhatsApp. Yes, sir. Thank you, sir. WhatsApp. Thank you very much, sir, for your wonderful message. And I now request Principal Professor N. Usha Garu to give a small message on this occasion, madam. Thank you. Over to you, Usha, madam.
Good morning to all of you. I invite all the research scholars, all the speakers, and the faculty members to this webinar. I congratulate the Department of Biotechnology and also Dr. Veer Brahmachari for organizing the first international webinar on translational cancer research. I do research persons, Dr. Gopinath and Dr. Bhattacharya would focus on the challenges in this area despite substantial progress, and I hope that they would address these challenges and tell us more about the advances which are going to come in translational cancer research. I wish and hope this seminar would help all of us in understanding these advances. Wish you all the best. Thank you very much for giving me this opportunity.
Thank you, Usha ma'am, for giving a wonderful message on this occasion. I would like to see registrar sir, is registrars are there? Professor Krishna Reddy Garu, sir. Sir, are you there? Hello. We'll wait for another 2 minutes, and I request all the participants to pick up your
90. This is the total number of compound 90, sir. This water OAP is 3.9, sir. This is whatever the active proton is the current proton to the 8.8. 8.2 is the to the Paris. The Paris content. This is the proton number of one more compound, disubstituted compound. This is the poly compound.
Naresh, can you hear me?
Okay. Shari, you can skip and
Naresh. Yeah, there is a lot. So somebody is speaking. Please keep your mute. Hello. Please mute yourself. Hello. Please mute yourself. There is a lot of background noise. I think Professor Krishna Reddy Garu is not there, and if you permit me, sir, I will go ahead with the presentation, inviting the guests. I cannot see all the people, so shall I go ahead with the presentation, sir? Hello. Hello. Seemana, are you ready? Seemana?
Yes, I'm ready.
Seemana, are you ready?
Yes, I'm ready.
Shall we go ahead, Seemana? And if in between, if I only registered, sir, is not yet come. So in between, if he joins, probably we'll ask him to give a small message on this occasion. Okay. So
Okay.
I would like.
Can you see my screen now?
Seemana Bhattacharya. Yeah, exactly. I can see your screen.
And you can hear me too?
Slowly, yeah, I can hear you.
Okay.
Well.
Okay.
Yes.
Okay.
Okay.
Yes.
Yeah, let me just introduce Seemana Bhattacharya. She has done her bachelor's in Bangalore University and master's from Mysore University. And I got an opportunity to work with Dr. Seemana in a laboratory at Indian Institute of Sciences, Bangalore, microbiology and cell biology. And she was there for a short span of a time. She worked as a project fellow with the same supervisor where I worked. And after that, she moved to Indian Institute of Chemical Biology in Calcutta for doing her Ph.D. under Mrinal Kanti Ghosh. She did her Ph.D. in cancer biology. And later on, she moved to Dallas Institute, where she did postdoctoral. And currently, she is working as a postdoctoral researcher in University of Texas Department of Leukemia, Houston. And she has contributed so much of science in the area of cancer biology. And I wish her all the best for her future endeavors. And Seemana, you can go ahead with the presentation. And probably you can go share the window and go ahead with the presentation, Seemana.
Okay.
Seemana?
Yeah, hi. Good morning, everyone. So,
yeah, go ahead, Seemana.
Thank you, Brahma, for the wonderful introduction. Yeah, it's shared, right? You can see my slide?
Not yet, Seemana. I think Gopinath is presenting. Gopi? Hello, Gopi? Gopi? Just now, registrar sir has joined, and we'll just, Seemana, just wait for a while, and registrar sir has come just now.
Okay.
We'll ask him to give a message. Registrar sir, are you there, sir? Registrar sir? Registrar sir? Professor Krishna Reddy Garu? Hello? Hello? Registrar sir? Registrar sir? We are just waiting.
I'm in online.
Okay. Sir, very good morning, Registrar sir, Professor K. Krishna Reddy Garu. On this occasion, on the occasion of today's international webinar, I request you to deliver a small message, sir. Thank you very much, sir.
Dear Dr. Brahmacharya Garu, the webinar convener. Really, we are very happy to note that KRISHNA University is conducting first time the international webinar in the relevant subject during the COVID-19. We really appreciate Dr. Brahmachari for taking a lot of interest and pains to arrange this international seminar within a fortnight. He has the dedication, passionation towards the research. I have noticed about 5 to 7 years ago, this webinar will give immense boost to our varsity, and also we are now having a roadmap to have 12B status and the NAC accreditation, and also to have the research center and RUSA funding. So Dr. Brahmachari contributions will be immensely beneficial to the system. And this webinar we are having for half a day, and the participants from the nook and corner of KRISHNA district and also Andhra Pradesh and elsewhere of the state will be beneficial, because the three experts are young and dynamic. They were really researchers who were born in India and shining in abroad. That is one of the crux of this international webinar. So these three eminent scholars will definitely give us impetus to the research in KRISHNA University, and hoping that this will go a long way to the Department of Biosciences, Biotechnology in particular. In general, KRISHNA University will be flourishing and nurturing under the able faculty of the which we are having in this institute. So thank you very much, Dr. Brahmachari, giving me this opportunity to give a small message and brief information regarding the international webinar. And I request Dr. Brahmachari to take over the proceedings. Dr. Brahmachari.
Thank you, sir. Thank you for a wonderful message, and thank you, sir. Thank you very much. And I now request Seemana to go ahead with the presentation, and just go and share the screen or the window. Yeah, Seemana, are you there? Seemana, yeah, go ahead and make the slide F5 so that it gets maximized.
Hello, can you hear me?
Yeah.
Okay.
Yeah, definitely, we can hear you.
And you can see my screen now?
Yes, we can. We are seeing your screen, and go ahead and maximize the screen, Seemana.
Yeah.
So you can't see anybody, but you're seeing the screen. Yes, go ahead.
So my screen is visible, right?
Maximize the screen, Seemana. Yes, go ahead. Yes, exactly. Yes, go ahead.
Okay, great. Okay, so good morning, everybody. Thank you, Dr. Veer Brahmachari, for the wonderful introduction. And first, I would like to thank the Department of Biosciences and Biotechnology at KRISHNA University and today's organizers, especially Dr. Veer Brahmachari, for inviting me to share my research experiences and work with the faculty and students. I truly wish today we can learn something new and make this webinar a great success. So like Dr. Veer Brahmachari just mentioned about my research journey, I did my BSc and MSc in biotechnology, and then I was in Professor M.S. Shaila's lab at IISc for a short while. And during this time, I was very fortunate to work under the guidance of Madam, and also to work with very talented scientists like Dr. Brahmachari and also Dr. Gopinath, who is our next speaker. After that, in CSIR, Indian Institute of Chemical Biology, I did my Ph.D. on basic signaling in cancer cells under the guidance of Dr. Mrinal Kanti Ghosh. And then, as I always had an interest in understanding more about the molecular mechanisms of cancer and how that can be utilized to create some translational work which has clinical applications. So I went on to do postdoctoral fellowships at Baylor Research Institute in Dallas and then at MD Anderson Cancer Center in Houston. And here I mostly worked on preclinical drug development and also translational oncology. So I will be sharing some of my data from both my PhD and postdoctoral work. So today's topic for my presentation is post-translational protein modifications in cancer. And so I will briefly touch upon the basic concepts of cancer and cell signaling. And then I will move on to post-translational modifications, and I will tell you why these are important and what are the different types of post-translational protein modifications. And then I will give some examples with which we can understand these mechanisms more in details. So to begin with, cancer, as we know, is an uncontrolled cell growth, is a disease of uncontrolled cell growth. And there is several deregulations in cellular signaling, which ultimately leads to aberrant cell division and cycling. So this imparts cancer cells with unlimited growth potential and irregular morphology, as well as altered metabolic capacity. So all this leads to formation of a tumor. So what is a tumor? It is a mass of cells arising due to abnormal growth in a tissue. So depending on the site of origin, a tumor can be classified as primary or secondary. And depending on their properties or aggressiveness, they can be classified as benign or malignant. So within a tumor, there is always histological and functional heterogeneity. So what does this mean? This means that a tumor mass is composed of different subpopulations of cells, while some are rapidly dividing cells, which form the mass of the tumor, while there are quiescent cancer stem cells, which have pluripotent or multipotent capacities and can self-renew themselves. So overall, what happens is a tumor cell and its environment, which consists of diverse immune cells and stromal cells and cytokines, it determines how a tumor grows or where it grows. So what is important to understand is there is a constant cell-to-cell communication, and therefore it is, Yes.
Seemana?
Yes.
Please maximize presentation. Presentation has, I think, it's in the small slide. Maximize.
It's in the presentation. It's in the max.
Yeah.
Can you see the whole slide now?
I think somebody else is also presenting in the presentation mode. That's why it's gone off actually. So
no.
Somebody else also came in as a presentation mode. That's why it's gone like this. So I don't know who is that.
Okay. What happened?
Somebody else came in the presentation mode.
What happened?
They started. That's why I think maybe Seemana, can you turn it off and turn it on, or something like log in, log out, or turn off the slide sharing and do the same thing again?
Yeah, just turn off and turn back.
Okay, I'll try that.
Turn off and turn back. Yeah.
Can you see my screen now?
Yes.
Yes, you can see now. Exactly. Yes, go ahead, Seemana. Yes, go ahead.
Okay, so you can see my whole deck now, right? Whole screen now, right?
Yes.
Okay.
Yes, we can see now. Go ahead.
Okay, so what I was telling is, from this, what we can understand is, there's a cancer cell or a tumor mass, and there are surrounding cells and cytokines or other associated cells and components, which among which there is always a constant cell-to-cell signaling or communication. This is known as cell signaling, and this is why cell signaling is so important. So what is cell signaling? Let's take a basic look at cell signaling. As you can see, it's a stepwise process, and it starts with extracellular ligands interacting with surface receptors, which leads to internalization of the signal. Once the signal is inside the cell, it can either show cytosolic effects or it can be transmitted inside the nucleus, where the ultimate effect is nuclear gene expression. So as you can see, this is a stepwise process, and we know that there are several proteins that are involved in this process. And all these proteins are subject to post-translational modifications. So now let's see what are post-translational modifications. Post-translational modification of a protein is a covalent modification, and it is carried out by specific enzymes. So how this occurs is there is a protein-protein interaction between the enzyme and its substrate. In this case, the enzyme is the protein modifier and the substrate is the target that is being modified. So the net outcome of a post-translational modification is a generation of a fully mature protein. So once a newly synthesized protein is formed, it is post-translationally modified to form a mature protein. Now, once it is post-translationally modified, a protein can undergo changes in structure, function, subcellular location, or even its fate. It's important to understand that post-translational modifications are very specific and they occur only on specific amino acid residues. As for example, a phosphorylation event will happen only on serine, threonine, or tyrosine residues, while modifications like ubiquitination, sumoylation, or nerylation will occur only on lysine residues. Now, these post-translational modifications can be reversible or irreversible in nature. And like any other enzymatic reaction, these have very high substrate specificity. So now let's look at what are the different types of post-translational modifications. Post-translational modifications are classified depending on the molecule that is added onto the target protein. As for example, a phosphorylation leads to addition of a phosphate molecule to the target, while glycosylation is addition of a carbohydrate moiety to the target. Similarly, acetylation, methylation are addition of acetyl or methyl groups, while ubiquitination, sumoylation, or nerylation is addition of ubiquitin or ubiquitin-like modifiers, which are sumo or ned8, to a target protein. Now, like I mentioned, these post-translational modifications are occurring by the help of certain enzymes or post-translational modifiers. As for example, when it's a phosphorylation event, a kinase is involved. When it is an glycosylation event, glycosyl transferases or glycosidases are involved. Similarly, for all these processes, there are specific enzymes which are involved. Like I mentioned earlier, that post-translational modifications in some cases are reversible. So there should be another set of enzymes which can do the reverse reaction. So dephosphorylation, deacetylation, demethylation, deubiquitination, these are the processes that happen to reverse the post-translational modification. And the enzymes that are involved in this case are phosphatases, deacetylases, deubiquitinases, or proteases. So what essentially happens is these enzymes hydrolyze the moiety that was added onto the target, thereby reversing the activity of the first enzymes. So there is one more factor that is very important to be understood over. So these post-translational modifications are very specific, but they are also interdependent on one another. So let's understand this by using an example. As for example, in the Wnt beta-catenin signaling pathway, beta-catenin is phosphorylated by the kinase CK1 at a particular serine residue. Once this happens, this phosphorylated beta-catenin is primed for the subsequent phosphorylations, which occurs by another kinase, which is GSK3 beta. And this phosphorylates beta-catenin at 3 other residues. Now, once this beta-catenin is phosphorylated on these residues, it is primed for ubiquitination by beta-TRCP and SKP complex. So you can see that this phosphorylated protein is tagged by a number of ubiquitin moieties, which is known as K48 linked polyubiquitination. And now this protein is ready for proteasomal degradation. So this is how one post-translational modification is related to another and is regulating another as a subsequent process. So next, let's look at the significance of the post-translational modifications overall. So once a protein is post-translationally modified, it can show altered protein structure, localization, as well as stability. And why all this is important is because this leads to diversification of protein functions. Also, all these reactions are very dynamic in nature and they are highly context specific, because of which they regulate cell signaling with respect to both space as well as time. It is known that post-translational modifications regulate almost all cellular events, such as gene expression, transport, signal transduction, protein-protein interaction, cellular communication, etc. And obviously, they are very frequently associated with developmental disorders and diseases, including cancer. So it is quite clear that post-translational modifications play a significant role in maintaining normal cellular state. So now let's take a deeper look at the processes of ubiquitination and deubiquitination among all the post-translational modifications that we just now saw. So the ubiquitin proteasome system was first identified in the 1980s, and it involves E1 conjugate, E1, E2, and E3 enzymes. So this is a stepwise process, and this leads to addition of a ubiquitin moiety onto a target protein. And this can be reversed by deubiquitination, where the deubiquitinating enzyme can cut the ubiquitin or hydrolyze the ubiquitin that is attached to a particular protein and reverse the ubiquitination on the particular protein. So now let's move on to our first example. So here I will discuss about the role of the E3 ligase each in the context of breast cancer. Now this example will help us to understand why a post-translational modification can be targeted. Because here we will see that itch is an important factor in maintaining the tumor microenvironment. And the reason for selecting this particular E3 ligase for working on it is it has immense implications in immune response. So first what we did is we took mammary gland tissues from mice. So we had 2 sets of mice, one which had wild type conditions and another in which there was whole body knockout of the E3 ligase each. So when we did a quantitative real-time PCR analysis of these tissues, we saw that there was increased pro-inflammatory signals and reduced tumor-promoting factors in the knockout, in the tissue that was obtained from the knockout mice. So what does this indicate? This indicates that there is reduction in tumor promotion when each E3 ligase is absent. So we hypothesized that each may have a tumor promoting role in this particular context. So further to test this, we injected breast cancer E0771 cells in both wild type and knockout mice, and then we monitored the tumor growth over time. So here you can look at the wild type and knockout mice, and you can look at the tumor that was excised after sacrificing the mice at the end of the experiment. So when we measured the tumor volume over time, you can see that the wild type mice, in the wild type mice, the tumor,
Seemana?
yes.
I think you may have to come.
Again, there is a presentation, same thing. Just go back and then. Yeah.
Okay,
someone
yeah, I'll do that.
did it, and I stopped it. Seemana, please go back. Yeah, please go back and do that. Seemana.
Is that okay?
No, not it. Please go back
Okay.
and just.
Okay, I'll do that.
Just go back, Seemana.
Yeah, I'm doing it.
Yeah, present now. Yes.
You can see my screen now? Can you see my screen now?
No, Seemana, not it.
Not yet?
Yes, it's coming. Wait a minute. Yeah.
Okay.
Yes, go ahead.
Okay. So,
Go ahead.
as I was mentioning, when we compared the tumor growth in the wild type versus the knockout mice, we can see that the wild type tumors, the tumors formed in the wild type mice were much larger compared to the tumor that was formed in the knockout mice. And this is a graph representing the growth kinetics over time. And you can see that the tumor volume in the wild type mice was really much higher than that in the knockout mice. And when the tumor weight was measured at the end of the experiment, here also we can see that there is significant decrease in the tumor volume when each is knocked out. So from this, we can clearly see that the E3 ubiquitin ligase each has a pro-tumorigenic role in the context of breast cancer. Next, we will move on to our second example. And here I will discuss about the deubiquitinase HOSPI. Now, this example will help us to understand the context-dependent roles of post-translational modifications. So, to initiate this experiment, we first did a large-scale screening to identify interacting partners of HOSPI. And in this screen, we found a very important partner which was interacting with HOSPI, and that is the retinoblastoma protein, RB. We know that RB has a very important role in regulating cell cycle, and it is a very important tumor suppressor. So then we delved much deeper into the mechanistic understanding. So for that, what we did is first, we generated two systems in normal cells. In one, we overexpressed HOSPI, as you can see here, and in another, we knocked down HOSPI using an shRNA. So here you can see that when HOSPI is overexpressed, RB expression level goes up, while when HOSPI is knocked down, we see that there is significant reduction in protein levels of RB. So at the same time, we also did quantitative real-time PCR to look at the RNA levels of RB, either in both overexpression system as well as in the knockdown system. So what we found is there was no significant change in the RNA level. So we see that there is a very nice correlation, positive correlation between RB and HOSPI expression in normal cells, but the RNA level remains same. So this indicates that the effect of HOSPI on RB is at the post-translational levels. Therefore, the RNA level doesn't change, only the protein level changes. This was further corroborated when we did a cyclohexamide chase assay to look at the stability of RB. So here you can see in the blue line, which shows that HOSPI is present. In that condition, RB half-life is extended much more when compared to the case in which HOSPI is absent. So this together shows that RB is post-translationally regulated by HOSPI, which leads to its stability. So this alone doesn't show that RB is a bona fide substrate of HOSPI deubiquitination. So to further study that, we did an immunoprecipitation reaction in which we used HOSPI antibody. And you can see that RB is identified as an interacting protein. And we also did a reciprocal immunoprecipitation reaction in which we used RB antibody. And we can see RB antibody, and we can see that HOSPI is interacting with RB. So this shows that HOSPI and RB are really interacting with each other. Further, we used immunofluorescent microscopy, and we can see that within the cells, we used two different cell lines. And in both cases, we can see that there is significant co-localization of both HOSPI and RB. Next, because this is a post-translational modification involving ubiquitination, so we set up a ubiquitination assay. So in the ubiquitination assay, what we do is we exogenously overexpress ubiquitin. Here you can see that we are overexpressing ubiquitin exogenously, and we are expressing the substrate, which is RB over here. And we are also expressing our enzyme, that is HOSPI. And CI over here indicates a catalytically inactive mutant form of HOSPI. Catalytically inactive mutant means it will not function like the wild type protein. So if we look at this lane, you can see that in the presence of exogenous ubiquitin, RB is heavily ubiquitinated. So this indicates ubiquitination of a protein. And when we provide HOSPI in this condition, the ubiquitination is significantly reduced. Now this doesn't happen when the mutant form of HOSPI is present. So this really shows that HOSPI deubiquitinates RB. So this is the first instance and that we found RB as a novel substrate of HOSPI-mediated deubiquitination. Next, we looked, because we were working on glioma, so we wanted to look at what happens in glioma with these two proteins. So we looked at different cell lines, glioma cell lines, and here actually what we found was surprisingly opposite to what we saw in the normal cells. So here you can see that where HOSPI level is high, RB level is low, while in the other cell line where HOSPI expression was lower, the RB expression was much more robust. Similarly, by immunofluorescence also, we were able to see that the cells which were showing high level of HOSPI expression had lower levels of RB expression, while cells showing lower level of HOSPI expression had much higher RB expression. We also analyzed about 20 patient samples from grade 4 glioma patients, and by immunohistochemical analysis, this is dabbed staining, and you can see the brown spots which indicate protein expression. So HOSPI expression is much high, while RB expression is not so much in the grade 4 patient tissues. So this indicates that there is a negative correlation between HOSPI and RB in glioma. But this was really very surprising. So the question we asked over here is, what is happening to RB in glioma? Again, we created two systems, one of HOSPI overexpression and another of HOSPI knockdown. But this time it was in a glioma cell line, U87MG. And in this case, again, we saw that when HOSPI is overexpressed, RB level protein expression goes down, while when HOSPI expression is knocked down, RB expression really goes up. So this again shows that there is a negative correlation between HOSPI and RB expression in glioma. When we did a similar ubiquitination assay, you can see that in the presence of wild-type HOSP, there is a lot of ubiquitination present on RB. So this indicates that there is probably a third player in this situation of glioma, which is regulating this. So we thought of a lot of players which may be involved, and we read a lot of literature, and we researched on this, and we came up with a lot of factors like P53 or P10 or AKT, which may have some common link with these two proteins. And ultimately, what we hypothesized is probably MDM2 might be playing a role. And why did we have this hypothesis? Based on literature review, we found that HOSP deubiquitinates MDM2. So MDM2 is stabilized in the presence of HOSP. And what we also found is RB is ubiquitinated by MDM2 for degradation. So looking at these literature, we hypothesized that might be MDM2 is playing a role as a third component in this equation. So to look at the role of MDM2, we again went back to analyzing the cell lines and the patient samples. And look what we found. U87MG with a high HOSP expression had high MDM2 expression, while DBTRG with a low HOSP expression had really low levels of MDM2. And again, looking at grade 4 patient samples, we saw that MDM2 is highly expressed in grade 4 patient samples. So all this indicated that HOSP and MDM2 had a very high positive correlation in glioma, while HOSP and RB was showing a negative correlation in the context of glioma. So to further see if it was really an MDM2 specific action or not, we overexpressed siRNA for MDM2, and in that condition we overexpressed HOSP. So you can see that RB level goes up only when MDM2 is absent. So when MDM2 is absent, the cell or the cellular context becomes more like a normal cell. So if we look at this in a schematic representation, we can see that in normal cells, RB level is high, and that is because RB is stabilized by HOSP. And in this situation, MDM2 is ubiquitinated and degraded. On the other hand, in glioma or in cancer cells, RB protein level is kept low. And how that happens is because HOSP is interacting with MDM2 and stabilizing it. And in turn, this MDM2 is ubiquitinating RB and sending it to degradation. Therefore, we identified HOSP as a novel deubiquitinase for RB, and we also show that MDM2 is a critical factor in case of cancer cells. So this gives us an idea that there are such kind of subtle differences between normal and cancer cells at the molecular level. And these are the nodes which have to be targeted for drug development. So to summarize my talk, we saw that cancer is an outcome of deregulated cell signaling, and how post-translational modifications are crucial in regulating these cellular signaling. And we also saw examples, one of an E3 ubiquitin ligase like itch, which provides a pro-tumorigenic microenvironment to facilitate breast cancer growth. While we also looked at another example, which is of deubiquitinase HOSP, and we saw how HOSP is deubiquitinase for RB, and MDM2 is a critical regulator of this deubiquitination in glioma cells. So by looking at these examples, we saw how post-translational modifications are important in cancer and how complex cellular signaling needs to be understood at the mechanistic level, and how protein post-translational modifiers can be targeted for drug development. We also saw several molecular techniques and model systems that can be utilized in studying translational cancer research. So I take this opportunity to thank my supervisors and mentors and all my colleagues for guiding me and helping me to do these work. And I have learned a lot from all of them. And as a researcher, what I have understood is there is no substitute for hard work, and we always have to keep working to strive what we aim for. Now, I would be happy to take any questions if you have, and I will exit from my presentation right now.
Thank you, Seemana, for your wonderful presentation. And,
Thank you.
Any questions from the audience? Any questions from the audience?
Can I start with that first?
Yeah. Please. Go ahead.
So the first story about the total, is it total body knockout of the itch? I mean, is it conditional or total? I can't hear you. You muted.
Seemana.
Yeah, it's a total body.
So it's viable. The mice are viable.
Yeah, but they are much smaller in size, and they're less immune compromised. I mean, they are more immune compromised.
Immune compromised.
Yeah.
And coming to the hospice story, the taking the 293T as the normal cell line, because it's a kidney epithelial cell line, and you're comparing with U87MG cells, which is a glioblastoma cell line.
Yes.
Wouldn't have been better if you've taken some other like Entera, like which can be differentiated into neural progenitors and things like that, or some kind of neural lineage cell lines, isn't it?
Actually, we did not use HEC293T. We used HEC293 cells. So the T cells are transformed. They have another viral oncogene in them.
That's right.
But yes, still HEC293 is also also a kidney cell. So although it's not exactly glial cell or neural cell. So if you look at one of my data, which is Western blot comparison between normal and between the different glioma cell lines, there I actually used NRA, which is normal rat astrocytes, and I used C6 rat glioma cells. So those were kind of controls. So if you work on glioma, you will understand that it's really very difficult to do any overexpression or anything.
I know they have very low transcription efficiency. We used to work on U87MG also. And also, we used to the cell line called Entera2, which we can differentiate into neural progenitors using retinoic acid treatment, actually. So we have to use lentiviral transduction. That I agree, definitely. Transient transductions don't work in those cell lines. So that's fine. I got your answer. It's fine, no problem. And using targeting these ubiquitination, it's a very broad pathway, right? So would it be specific to cancer cells? How much side effects you would think it will happen?
Yeah. Targeting ubiquitin system is, there is bortezomib, which is a proteasome targeting drug, which is widely used in the leukemia space. So actually, targeting proteasome system as a whole is not going to work, but targeting particular ligases or deubiquitinases, actually there are a lot of inhibitors. And a lot of people are working on HOSPI inhibitors. So those will actually work, and they will have much lower non-specificity.
Okay. So, nice talk. Thank you so much.
Definitely, it is very important to understand the context. That's why in the second example, I showed the difference between normal and glioma cells. So we have to really understand the context before targeting.
Agree. Yes, sure. Yes. Thanks a lot. Nice talk.
Thank you.
Yeah. Any other questions from the audience? Any of the colleagues from the department and other people, other departments, other universities and other colleges? Any questions? Okay. I have a small question, Seemana.
Yes.
So what was the, just now Gopinath was mentioning about the lentiviral transfection system. So what was your transfection system for infecting the glioma cell lines?
Yeah, we use a lentiviral system. For the SH, for the knockdown, we used the PLKO vector system. And for overexpression, we used neomycin cassette in a lentiviral vector system so that we can select the cells later on.
Okay. So what was the actual hypothesis to end up in locating MSP2 as a proteasome complex degradation?
Sorry, I couldn't hear.
MSP2. You said you hypothesized MSP2 complex for the degradation of proteasome complex.
No, MDM2, I see.
Yeah, MDM2 complex.
Yeah, so actually what happened is because HOSPI and RB were behaving entirely opposite in the glioma cells as compared to the normal cells, we were kind of stuck at that point and we did not know what was happening. So we started to read things and we also, I did not show the data, but we also did these experiments in different P53 backgrounds. So we took P53 null cells, we took P53 mutant cell lines, P53 wild type cell lines, P53 heterogeneous cell lines, and we looked at the correlation between HOSPI and RB in those cell lines. And there we saw that P53 background did not do anything at all. So there is actually quite a bit of literature of HOSPI, P53 and MDM2, because this protein HOSPI was initially identified as a deubiquitinating enzyme for P53. And then later it was shown that it also deubiquitinates MDM2. So both P53 and MDM2 are substrates of HOSPI. So we thought that if P53 is not playing a role, we thought, let's see if MDM2 is playing a role. So in that, that's how we ended up in looking at P53 and MDM2, and P53, and this process came up to be P53 independent. But when we looked at MDM2, we also did more experiments. I did not show it here. It's there actually in the paper which we published in Febs in 2014. So there we show with different techniques that when MDM2 is present, then HOSPI and RB regulation is different. But when we knock down or use an siRNA to remove MDM2 from the system, then HOSPI can actually go and work on RB. All the other time when MDM2 is present, HOSPI is sequestered, functioning with MDM2 or stabilizing MDM2.
Thank you very much, Seemana. And any other questions to be taken from the audience? I'm waiting to take up any questions. Hello? Okay, Seemana, because there's a large crowd of students, student fraternity who is listening to this lecture. Nearly there are nearly around 600 to 700 students from across 7 states in India. Okay, it's a very large meeting. And as of now, kept a part of your technical meeting and technical session on cancer biology. So I request you to give at least 1 or 2 sentences on how did you grow up till your career from your master's to a very good postdoctoral scientist in cancer biology. A small message to the students, Seemana.
So I would say thanks to all of you who were present in IISc lab. And as I mentioned earlier, my IISc lab experience was my first research lab experience. And I was very fortunate to be trained by Dr. Gopinath, who is our next speaker. And I am also I learned a lot of things from Dr. V.R. Brahmachari and other fellows who were there in the lab at that time. So that's how I was motivated to hang on to PhD. PhD is something which requires a lot of tenacity. So all these people who, some of them were working for their PhD and some of them were already PhDs. So they motivated me. And of course, a lot of my mentors, including my professors in college, in BSc, in MSc, they all motivated me. In my MSc, I was always told that you have to qualify for CSIR net, and that's the only way to go. So yes, I was kind of one-track mind, and I pursued that. And luckily, I got through the net, and then I went for PhD. And during my PhD also, I had a lot of very hardworking colleagues. So I mentioned already, there is no substitute for hard work. So keep working. Don't think you are losing it. You have to keep working. And there will be times when you will really feel that, oh, my PhD is not going anywhere. But once you are on the path, you will end up doing a PhD. But to make it significant, you have to be into it with your entire body and mind. And if you really think you want to do something in future, you have to keep pursuing that goal and you have to keep yourself self-motivated. I hope that encourages students.
Okay, thank you, Seemana, for a wonderful presentation and a wonderful message to all the students. And
thank you.
Thank you, Seemana, for sharing a valuable time. And really, we are lucky to have you today for delivering a lecture on cancer biology in advance.
Thank you.
And
thank you.
Hoping to
thank you for giving me this opportunity.
Hoping to see you deliver. Thank you for we would expect some more lectures from you to our students especially. Okay. Thank you, Seemana. Good day.
Thank you.
I now request, please unmute.
Okay, I will unmute.
Yeah. Dr. Gopinath.
Yes, I'm here.
Dr. Gopinath. Gopinath. Yes, fine. Let me introduce the next speaker, Dr. Gopinath Sundaram. So it's very fortunate to work with Dr. Gopinath in the same lab and the same supervisor at the Institute of Microbiology and Cell Biology. So very cheerful person, very affable academician, very hardworking individual, and we are all fortunate to have you, be as a laboratory colleague with IISc, and comes to his education. He's done his master's in Pondicherry University and PhD in Inaugural Sciences in molecular virology, and he worked as a scientist in postdoctoral scientist in the Institute of Medical Biology in Singapore for several years. And very recently, he has joined as a principal scientist in CFTRI in Mysore. And hello, one second. One second. Let me just. I'm removing one person. Yeah, 1 second. Okay. And when it comes to Gopinath's publications and CV, it's just 3 pages CV, very short CV, Gopinath. And let me tell you, he has got 2 patents and only 19 publications. Out of 19 publications, 2 Nature Communications, 1 Nature Medicine, 1 Nature paper, Journal of Experimental Medicine, 2 papers, and Infection Immunity, BMC Genomics, Cell Report, Molecular Cell Oncology, FEBS Web Journal, and all are very, very, very high impact factor journals. We are very fortunate to have you today, Gopinath. Okay, so and I'm very fortunate to have you, for me to work with you along with you at IISc. And I now request Dr. Gopinath to come out with the presentation. Gopinath, go ahead.
Yeah. Thank you, Dr. Brahma and Dr. Seemana. as he mentioned that we did work together, and those times. First, let me talk about Seemana. She was really hardworking student. In fact, in my tenure, we had about 12 students, out of which she was the most hardworking, I could say. In fact, even in December, she used to sweat and work. I used to see her actually. And Dr. Brahma is another vibrant fellow, always cheerful. In fact, it was very nice to have him in the lab, and amazing time we spent together. So yeah, that time the PhD was in virology, molecular virology specifically. So today's talk, what I plan to do is, okay, before going that, actually, we used to work on viruses which are about 200 nanometers in size. So from viruses, once I went to postdoc, I totally switched to micro world, otherwise known as microRNAs. And after coming to CFTRI, I went back to nano world, working on nanoparticles. So what I'm going to summarize today is the work of last 12 years in 2 different parts in about 45 minutes. And also, if time permits, I will just briefly touch upon some COVID-related work we have started. So I think if time permits, okay? So let me start sharing the slides first. So I should do it, okay, present now. Your entire screen, okay. So let me go to screen, yes. Okay, so can you all see my screen, right? Okay, fine. Okay, fine. So before I get into the talk, let me first acknowledge the people who was part of this. So my postdoctoral work, I was in Institute of Medical Biology in 2009. And this is my mentor, Dr. Prabhas Sampath, and we had a lot of collaborators. And the work I'm going to present today was done by me and Dr. Srikanth Lama, who is also from IISc, Dr. Mohsen, who is also from IISc, and Dr. Manish and Mr. Poyok and Mr. Hisham. And this is the group we had in between 2009 and 2018. And this is my current lab. So I have about 3 PhD students, and the work I'm going to present today is done by this very talented, very hardworking student called Mr. Sriram Peringattu, that's his full name, okay? Okay, so the title of the talk I've kept is Wound Healing, Cancer and Non-Coding RNAs. You would all probably be wondering why wound healing and cancer. Is there any link between these two? Because we all get injury in life, like, when you shave, suddenly you get a cut, nothing happens actually. But it turns out tumors, yeah. Yes.
PPT, put the PPT on, yeah.
Sorry?
Yeah, it's come now, yes.
Now it's visible?
Yeah.
Okay.
Maximize.
Okay. It's fine, right?
Maximize it and go ahead. Maximize it and go ahead.
It's already maximized.
It's still in the slide mode, and maximize it and go ahead. Yeah, go ahead.
Okay. So why wound healing and tumors are related? In fact, it has been predicted in almost about 4 decades ago that tumors are considered to be wounds that do not heal. Okay. So if you look at several pathways and several growth factors, like you talk about any pathway involved, yeah?
Gopi.
Any problem?
Gopi, full screen. It's not in full screen, Gopi.
Is it fine now?
Type this, maximize the screen. No, Gopi.
Yeah.
It's still in the style mode.
Oh, it's in slide mode. So I'm trying to make it full screen.
The large icon here. Is it slideshow? Go on the slideshow.
It's on the slideshow right now, actually.
That one, yes.
It's fine now?
I don't know, somehow. No, click that one.
Let me do that again. Probably
Yes.
I'll remove this guy.
It's still in the trial mode, Gopi.
Is it better now? You can see it now?
Maximize.
Okay, let me try.
We can see it.
Oops. Because when I go slides share.
Is it Mac?
It's Mac, yeah.
Is that Mac?
Yeah, it's Mac.
Okay. So, how will Skype F5?
Okay, let me try F5. F5 is function F5. No, it doesn't work. No. But
the screen is somehow very small.
My screen is very small.
Click only one slide and go ahead with the slideshow.
You can see now?
No, not visible clearly, Gopi. not visible, but I think you
Not visible.
need to go for slideshow, because Gopi, in Google Meet, what happens is you don't see the people, only you see your own screen. So in that case, go ahead and directly shoot.
You may go to slideshow.
Slide screen.
I try. It's already in slideshow.
Yeah,
right?
now it's come.
Now it's come.
Now it's come. Yes, go ahead, Gopi.
Okay,
And
so let's just go.
maximize the screen.
It's in the slideshow mode now, actually.
Yeah,
Okay.
go ahead.
Fine. So just to take the crisp message and the take-home messages, there is always a link between wound healing and cancer. In terms of the processes involved, like angiogenesis, cell migration, cell proliferation, or in terms of the growth factors involved, like EGF, TGF-beta, FGF, everything is linked. Okay. And if you look at the cell types also, like macrophages, we call them tumor-associated macrophages, neutrophils, tumor-associated neutrophils, stem cells, cancer stem cells. So everything is linked, which is there in both wound healing as well as in cancer. So does it mean, since I said cancer is a chronic wound, but can injury cause a cancer? Sometime in 2016, I was running. So suddenly I fell down. So I got an injury like this. This is day 2 post-injury. Day 31, it's fine. I didn't have a cancer. 2 things I learned from this. First thing, injury itself do not cause cancer. Second thing, while jogging, do not look into your phone. Okay. So, but there seems to be a tremendous link between injury and cancer. That's the crux of the story. So the top contents is basically, the first part will cover a little bit about wound healing and how exactly non-coding RNAs are there in wound healing, and how we identified some kind of non-coding RNA mechanism in wound healing, and how is it linked in cancers, because cancer seems to hijack some of these processes in wound healing, and what is the cause, what is the consequence, and how can we exploit this molecular switch by non-coding RNAs for drug discovery. After this, in second part covers some edible nanoparticles for wound healing in cancer, which we started recently in the lab. Okay, and a bit of COVID work. Okay, so when I went for my postdoctoral work, my work was to look at skin. We all take our skin for granted. In fact, skin is the largest organ in your body next to intestine. In fact, if you go for, water molecule is about 18 Daltons in size. Let's go for swimming. You go for swimming, even water does not penetrate, you don't come out swollen, right? Which means the skin is doesn't let even the water molecule to penetrate. That means it's giving you the barrier function, keeping you in sterile. And that barrier function is conferred by the topmost layer of your skin called epithelium. Epidermis right here. You can see this epidermis, then you have the dermal structure, then you have fat, you have hair follicles. This topmost layer called epidermis, which is less than a paper's thick, is the one which gives you the barrier function, which does not let anything go inside. Okay. And if you look at this, it's a regenerating tissue. Okay, it has one proliferating layer called basal layer, which proliferates and then differentiates, into multiple layers. Let's not get into the details. And then ultimately you keep the dead cell comes up called a stratum corneum, which we keep shedding every day. And it's a process just like a lunar cycle. For a cell to migrate from here to there to reach this stage, it takes about 28 days. If you do not have skin, let's say you somehow get burned or something, this is what happens. You literally can get fried and you can get dehydrated. Water can just simply escape from your body. So such an important organ of the skin. What happens when you have an injury? Got it? So since barrier function is very important, our body has this very sophisticated mechanism of healing wounds. When you have an injury, there's a lot of process that kicks in. As you've probably noticed, the first thing what happens when you have an injury is a blood clot, which happens within minutes. We call them hemostasis or coagulation. This process starts within minutes, and then the second process is inflammation. Yes, it also starts within minutes, and it can go on for weeks. That's what gives you pain and all those things. But the most important process in wound healing is as I mentioned before, the migration and proliferation of skin cells. As I mentioned before that I said you have one layer of proliferating cells and then everything else is differentiating. They are post-mitotic. But in skin, what happens when you have an injury, you want to seal the break. So to do that, these proliferating cells start hyperproliferating, and the cells which are not used to migration, which are not used to movement, they start moving over the wound. You see the cells right here going around on top of the wound? This is a very important process. This starts within 6 hours post-injury. Okay, so then you have this remodeling phase. You end up with scars because you try to get back the normal architecture of your dermis, which has collagen fibers, but you don't get it usually. But then you end up with a scar tissue. But why do we have to study this? Why do we have to study about wound healing? Okay, fine. It's not that we take it for granted actually. But think about people with chronic non-healing wounds, especially diabetic ulcers. Probably would have seen people with chronic wounds. Usually, often what happens is in these patients, the wounds do not heal for a very long time. Because in a normal healthy person, wounds heal, we take it for granted. But in those diabetic patients, when a wound comes, 6 weeks, 8 weeks, wounds don't heal, they get infected, you have to amputate the extremities. Almost 75% of cases in diabetic ulcers do not heal. Out of the 75%, 25% leads to amputation. Got it? So if you understand what is important for wound healing, you can practically define what is not happening here. That's the reason we will try to understand wound healing. Okay? So in that process, we try to look at non-coding RNAs. So this is where non-coding RNAs come to this. A specific class of non-coding RNAs. Because non-coding RNAs is very broad. For example, ribosomal RNAs is also non-coding RNAs. But we are particularly focusing on a population of non-coding RNAs called as microRNAs. And as Dr. Seemana was talking about post-translational modifications, I'm going to talk about post-transcriptional. Because once you make the protein, by phosphorylation, ubiquitination, you can decide whether the protein is going to be functional, it's going to be there or not. But even before making the protein, when you have the mRNA, you can still have some kind of regulations there using these microRNAs, which can decide whether the mRNA is going to be there or not, whether it's going to be translated or not. So that is the function of microRNAs. This is a very short 20 nucleotide size, and they regulate more than 1/3 of the genomic output. And they have this own things like one microRNA can target many mRNAs, and one mRNA can be targeted by multiple microRNAs. And they were discovered about 25 years ago, but they've already entered into clinical trials. So to make this microRNA, as I said, it's very small, right? Like 22 nucleotides. And this is the basic biology. Everything, any RNA produced in the cells has to be transcribed. Got it? So even microRNAs are transcribed, but they're transcribed by same RNA polymerase 2, which is making your mRNAs also as a long transcript, because RNA poly2 cannot make short transcripts. It's not used to making short 22 base pairs. No, it makes a large primary transcript, which is also similar to mRNA in terms of having a cap structure, poly A tail, but it also has the stem loop structure where you have the mature microRNA sequence. So let's take this. This is called as primary microRNA. Just keep this in mind. Primary microRNA is similar to mRNA. You have the mRNA, the precursor sequence right here. Then it is cropped inside the nucleus, just the stem loop. Then the stem loop comes out in cytoplasm, and then it's further cropped. We call them dicing. Then you have these two molecules, two microRNA molecules, complementary molecules, and selectively one molecule is stripped off. Then you have the mature microRNA, about 20 to 22 bases in size, which will bind to the target mRNA due to complementarity. Watson quick base pairing, and leads to translational repression or mRNA degradation. And within this, if you classify these microRNAs, that as I mentioned, that one primary microRNA can have one microRNA. In fact, what's happening is that these are called as intergenic. That is how exactly cells are making these microRNAs. Wherever you have space between genes in the genome, they are used for making these microRNAs. They are called as intergenic, between the genes. Got it? So if you realize that you're spending a lot of energy in making a small transcript, so what happens sometimes, you can have multiple microRNAs in a single primary transcript. But to be much more intelligent, what you do is at least 50% of microRNAs are made from the introns. Introns, it's not getting used for making the mRNA. So you take the introns to make microRNAs. Right? I've got a couple of examples here. You have this intronic microRNA, couple of examples, which means it's a very brilliant system because you use energy conservatively. But in few cases, less than 5%, we have a very interesting,
Gopi, I have a small thing. Gopi?
Yeah.
Just check, because slide is not moving, or rather it's not getting maximized and we are unable to see. Have you gone through first slide or still in the first slide, Gopi?
No, I've gone through almost 5 slides actually.
Oh, see, we are seeing only first slide, Gopi, so we are unable to see.
Oh, is it?
Yeah. There is some problem with your Mac.
Let me log out and come back.
Okay, now okay.
Just go back and do that.
Now microRNA classification slide is displayed.
Oh, sorry. Wait a minute. I think the problem could be that when I go to full screen, it is. Now is it seen?
Just go back and do it again, Gopi. Just go back. Because the slide is not moving from your Mac.
Now it has moved.
No, Gopi, just go back and represent.
Okay, let me try doing that actually.
Yeah, just go back.
So now can you see this slide moving? I'm moving the slides now. Can you see?
Yes, moving.
It's moving, right? Okay, I think just a quick summary is I was still talking about how exactly microRNAs are being made. They made as a long primary transcripts called primary microRNA, similar to mRNAs. Then this gets cropped inside to make the precursor microRNA. Then this comes out and this head is removed. Then you have this binary structure. Then it's kind of one strand is selectively processed and that comes out that binds to target mRNA. Got it? So if you look at the types of microRNAs in terms of their origin, so what happens is wherever you have gaps between genes, you use them to make this microRNA. For example, you have this primary transcript mRNA structure here with the microRNA, and these are called as intergenic microRNAs. And then you can have multiple microRNAs in one primary transcript. These are much better option because you spend less energy. Then you have intronic microRNAs where you use introns to make microRNAs. Is it still okay, clear? Yeah. So this is an economical solution for cells because it's better to use the waste to wealth. That's kind of approach cells are taking here. So you use introns to make this microRNA. But in few cases called as exonic microRNAs, you use exons. When I say exons, you have to be really careful here. An exon means it's a functional part of your mRNA, which means it could be 5' UTR, the open trading frame, and 3' UTR. The problem is that if an mRNA is making microRNA, obviously it has to be chopped off into pieces. So that is the crux of the story. Okay, so our lab is primarily looking into microRNAs in wound healing. Like how exactly? I was going back to the non-healing wounds. So we want to understand the microRNAs and their role in wound healing. So how we do it? So obviously we don't want to get into mouse. So the slide is moving now, right? You can hear me? Slide is moving?
Yes.
Okay, fine. So how do you do it? How do you create injury in the laboratory? We don't want to go for mouse because mouse skin is not close to human skin. Mouse does not get psoriasis. Mouse has so much of hair follicles. We don't have that much of hair follicle density. So that's not really similar to human skin. So what do? We go for hospitals and getting clinically discolored skin. So we get this because people with big tummies or post pregnancy, they want to get rid of their excess tummy. So they get the skin along with the fat. So they get rid of the fat right here. And you see this triangle right in the center. So that we use for creating injury. So what we do is very simple. We just make little biopsies here. We avoid the corners because corners are already injured. So we just make a biopsy in the center like this, the small donut biopsies, and don't put them in the medium, just expose them to medium just from the bottom. It just to simulate the skin because your skin do not get blood supply in the top. So to simulate that, we call the system as air liquid interface. So where the medium is just supplied from the bottom. So right after making a biopsy, either snap freeze it in liquid nitrogen, which we consider as an uninjured skin, or grow it for 24 hours, or like this, and see what happens, how exactly injury proceeds, and see what microRNAs that change during this process. Okay? Yeah. So in this process, so as I said, we are looking at microRNA. So what we did was we isolated RNA from the either 0 hours post-injury or 24 hours post-injury and did a microarray, simple microarray for the microRNAs. And then we looked at, we had several microRNAs. I'm going to talk about just one of them. It's called as microRNA 198. We see that this microRNA is expressed very highly in the epidermis. You see right here, red color. The red color intensity is for in situ hybridization for the mature microRNA. And this microRNA is gone in the cells, which is, you see this migrating tongue right here, right? You see that. So these are the migrating tongue, the cells which have started migrating. So the microRNA is completely gone. So maybe this microRNA inhibits the migration of cells. That's the inference from this, or probably we also confirm that, as you can see here. So if you overexpress this microRNA in the cells, what happens is that this microRNA inhibits. You can see that this is Scratchman assay, where cells migrate pretty fast from control cells. If you express a lot of this microRNA 198, you see they are migrating slower, which makes sense because this microRNA inhibits migration. It is highly expressed in normal skin. Because in the normal skin, you do not want this migration of skin, so you have this microRNA in place. But things became really interesting when we realized who is making this microRNA. That is where we realized that, the animation is kind of gone. Okay, fine. That is where we realized that this microRNA is a part of this gene called follistatin-like 1. It is present in the 3' UTR of this gene. It is an exonic microRNA. Got it? If you remember correctly, that exonic microRNA, they have to make a decision. Like, if you want to make the microRNA, you have to chop off the mRNA into pieces, or if you want to have the protein, what happens with protein? You see this? Okay, so let me just do that here. So in this particular case, if an mRNA is making the microRNA, mRNA is bound to make proteins, right? So what will happen is if you want to make the microRNA, you have to break it into pieces. If you want the protein, you just want to keep it as such. So in this particular case, what happens to the fate of the protein? So question is whether this FSL mRNA which is coding for this microRNA, is it a non-coding RNA or is it a coding mRNA? So this is where we realize there is a molecular switch. A single mRNA switching between two fate decisions. So as you can see here, at normal skin you have lots of this microRNA. Upon injury, you see this protein start expressing. The microRNA is not there. So just remember that just remember that one single mRNA switches between two fate decisions, one for microRNA, one for protein, depending on the physiological state. And they have opposite functions. We have realized that the microRNA suppresses migration, whereas this protein, which is coming out after the injury, is required for the promoting the migration of skin cells. So I think I'll switch this. I don't want to get into this again. So to cut a long story short, this is what happens. So in the normal skin, you have this mRNA getting into microRNA biogenesis. So you have this microRNA there in the normal skin because it doesn't want to make the protein FSTL1, which is right here, and also another target of this microRNA, diaphanous-1. So let's talk about just these three molecules. Diaphanous-1, which is an actin regulator, which is required for migration. So you don't want the expression of diaphanous-1 and FSTL1 in the skin so that you express microRNA. Upon injury, the same mRNA comes out of cytoplasm, does not get processed into microRNA, comes to cytoplasm, gets translated, and it makes the FSTL1 protein, which is required for migration, along with the target of this microRNA, diaphanous-1. So you have two proteins now, okay, which promotes migration. So this is the molecular switch I was talking about. And most importantly, when you looked at diabetic ulcer patients, like, the wound edge samples from diabetic ulcers, the picture I showed you before, what you see is this switch is defective. Actually, even though they have so much of injury, they have so much of hyperproliferation. You see a pretty thick skin right here. They fail to turn on the switch. They express loads of this microRNA 198. They do not turn on FSTL1. That is one of the reasons the skin cells in the diabetic ulcers do not migrate and the wounds do not close. So this story was kind of published in 2013. So I don't want to get into the details how exactly this process is regulated. Basically, what happens is you have it's regulated through RNA binding protein, which decides whether the mRNA is going to go for microRNA processing or as an mRNA. And the upstream signal, you have this TGF-beta, which controls this protein called KSRP. Okay, fine. So at this point of time, we realize, okay, it's very nice that you have a very good molecular switch specifically dedicated for controlling the migration just by single mRNA. But coming back, okay, just to think to remember that it's like, having this switch is kind of, so in the normal skin, you have this FSL mRNA. Oops. I think I'll skip this. So in the normal skin, you have this FSTL mRNA, which it's like a Monday morning. So it's making only microRNA which suppresses diaphanous one. So practically you don't migrate. Whereas upon injury, the injured skin, they switch. They start making FSTL1, they downregulate microRNA 198. So you're also expressing diaphanous one. So now you kind of very active, the skin cells start migrating. This is regulated by TGF-beta, and then in chronic ulcers, this doesn't happen, so they don't migrate. Oops. Now we'll come to the cancer part of it. So as I mentioned before, that cancer is tightly linked to wound healing. A lot of processes are similar between cancer and wound healing in terms of the processes like angiogenesis, cell migration, but not in the form of migration, but we call them invasion and metastasis. Migration is like walking, invasion is like swimming, because invasion you have a resistance. You have to go through the extracellular matrix, cells have to go through. Whether migration is just walking on top of your extracellular matrix. Okay, so we ask the question, okay, if cancer and wound healing is tightly linked, by any chance is there any link between cancer and wound healing via the switch? So the reason we focused is because there was a lot of cases of head and neck cancer coming up in Singapore. I used to work in Singapore at that time. And we had about 4,500 deaths due to cancer, and at least 300 of them are due to head and neck cancers in Singapore. So there was a lot of stress on working on cancer. So we asked the question, what happens to this molecular switch in head and neck cancers? Because skin cancer is not at dangerous for anybody in Asian countries, because we all have a lot of melanin pigmentation, so we won't get that much of skin cancer. But head and neck cancer is quite prominent. And if you look at head and neck epithelium, is quite similar to skin epithelium. Some minor differences are there. So we looked at the switch. What we did, we took normal tongue section, normal skin we already had, and head and neck cancers. What is happening is head and neck cancers also behave like they've been injured. That is, they turn off the microRNA 198, they gain the expression of FSTL1. Since they turn off the microRNA 198, you also gain the expression of diaphanous 1. That is what is happening. So they have stolen the switch. Sorry. So if they've stolen the switch, why did they do it? So is it required for migration of cancer cells? In cancers, you have these two steps, especially in epithelial cancers, because 90% of cancers are epithelial cancers, and rest of them are lymphoma and glioblastoma and all those things. In addition to that, 90% of cancer death is due to metastasis. That is, as long as you have a primary tumor, let's say you have a big skin part of skin tumor, as long as it's there, nothing would probably happen to you, just that you become weak because they're sucking up all the nutrients, what you have, what you eat. But the death is due to these cells not staying there. They start invading, getting into the blood vessel or lymph nodes and getting into the vital organs and colonize and affecting the vital organs functions, like your liver, like your lungs, like your bone marrow. So death happens due to metastasis where vital organs are involved. So more than the cancer itself, you have to stop the metastasis. So probably, since I talked about cell invasion, this switch involved in cell migration, could it be that they use this molecular switch for this particular process? So that's what we want to test. And cell migration means you could probably do a scratch wound again, but that's not physiologically relevant. So we wanted to have a physiologically relevant model to mimic the step one. That is, first step is to the cancer cells to invade through the basal membrane into the extracellular matrix. That is the first process. Second process is once they are in the blood, they have to get into the vital organs and colonize. So we simulated these two steps using two different model systems. The first step can be simulated using something called as organotypic model system. So basically, what is an organotypic model? It's a 3D skin. Like 30 years ago, we developed, people have already developed making 3D skin in cultures. Basically, skin is nothing but your dermal tissues, which is about 2 millimeters thick. On top of it, you have this epithelial cells, the skin cells, which is making probably 5 to 10 cell thickness. So in this model, what happens is you just use collagen to reconstitute your dermis and just put some fibroblasts so that they remodel the collagen so your epithelial cells can sit on that. And then you see keratinocytes or skin cells on top and expose them to air for about 2 weeks, 2 to 3 weeks. You get a very nice skin like this, like just like your epidermis, just like your normal skin. But in this model system, if you see cancer cells, or instead of normal skin cells, you see cancer cells, they are not used to sitting idle here. So what they do, they start invading down in the matrix. This happens over a period of 3 weeks, not like cell migration, which you just observe in 1 or 2 days. This happens over a period of 3 weeks, exactly mimicking the step 1, whatever I mentioned before. So using this model, we check what happens, what is the role of these two genes which we gain by the switch, that is FSTL1 and diaphanous 1. So what we did, we take cancer cells and knocked down these two genes, FSTL1 knockdown, diaphanous 1 knockdown. This is the control cells. You see control cells within about 2 to 3 weeks, your cells have invaded down, like about I think probably about 100 or 200 microns they've invaded. If you knock these genes down, then they do not invade, which means both genes, by turning on the switch, you occur these two genes, FSTL, you turn off the microRNA 198, you occur these two genes. They are required for this first process to cross the base membrane and go into the dermal matrix. How about the second step, colonization? What we did was you inject the cancer cells into mice. Okay. Sorry. The second step, what you do, simply inject cancer cells into the tail vein of the mice and leave for 2 weeks. Usually what happens, the cells, it's like a second step. The cells will get into the bloodstream, go to organs wherever you have lots of blood vessels. One of the primary organs is your lungs. So we label the cells with a vital dye or luciferase, so we can visualize it without killing the animals. So this is the control animal where this is day 2 post-injection of cells. You can see the signal is in the lungs, and 25 days post-injection you'll see lots of it become red in color because the cells are really actively proliferating and colonizing in the lung. When you knock down these genes, you see this diaphanous 1 knockdown, FSTL1 knockdown, or combined knockdown, you don't see anything, which means acquiring this molecular switch and turning on these two genes, FSTL1 and diaphanous 1, is required for both processes. One is the primary step 1 where you double invasion, as well as going and colonizing in the target organ, which is in this particular case is lungs. We also saw the same thing in patients, same thing. If you just take patient samples and just look at the expression level of these two oncogenes, FSTL1 and diaphanous 1, classify them based on high expression. The patients who has high levels of these two oncogenes, patients having only high level of only one of them, or none of them. Okay, so if you classify them, just to summarize, you see that the green line here, these are the patients who have very less levels of both genes. These two lines are the ones which are having either FSTL1 or diaphanous 1. They die much faster. But when you have both these genes together, the probability of survival is very less for you, which means acquisition of this switch decides how long you're going to survive. Got it? So this is what we publish, that it's a complex mechanism how exactly these two things happens. But just to take home message is that in head and neck cancers, they hijack the switch. They hijack the switch to promote their invasion and metastasis. Okay, so how do we exploit this switch for drug discovery? So that's what is the third part of my talk. Okay, I'm going. Okay, fine. How do we exploit the switch for drug discovery? What we realized is that when we looked at patient samples, like we used to buy this tissue arrays which used to have hundreds of patient samples. What we realize is that when you correlate the expression of microRNA 198 with FSTL1 and diaphanous 1, whichever patient sections had high levels of microRNA 198, they had very little levels of FSTL1 diaphanous 1. Which means if there's any way you can induce microRNA 198 in patients, then you get rid of two oncogenes. Can we do that? So how do we do that? That's the crux of the story. So how do we induce microRNA 198 endogenously? Can we screen for drugs which can use this microRNA? To do that, we developed a sensor. Sensor is nothing but it's basically what we do. We make a construct which has a promoter like called EF1 alpha and a GFP. MOTC is the one which kind of decreases the half-life of GFP. Don't bother about it. And then we insert microRNA 198 binding site in about 10 binding sites in this construct. For normalization, we have this M cherry which is red in color. So let's say a drug and we make a stable cell line out of it. So we using lentivirus, we use this construct. A cell will be constantly expressing this mRNA. Now let's say if we add a drug that induces microRNA 198. What will happen? The 198 will go and bind to this target binding sites. which will lead to decrease in GFP. Got it? But nothing should happen to MCHERI. Maybe let's say that a drug induces toxicity, then both MCHERI as well as GFP will go down. Right. Whereas if it is because of microRNA without much of toxicity, then only GFP will go down, MCHERI will not go down. So that's what this is a proof of principle. Like this is the control cells having both green and red. You add more of, you see, this is the dose difference. When you transfect these guys with microRNA, you see the GFP is disappearing. Higher concentration, no GFP. As you go down in concentration, that GFP is coming back, which means the system is kind of working. It's kind of very sensitive for induction of microRNA. Got it? So, using this system, we do a drug screen. We took about 978 FDA approved drugs and do a screen. So, this is a system we use. This is a high throughput imaging system wherein we can put 384 plates and 96 plates and just do a image. We treat with the drugs and that will image every well, probably about 50 images per well, so we can have more statistics in that. So this is a typical image looks like. So these are the different drugs we have treated, about 96 out of the 978 compounds. But the end result is that if you plot GFP by MCHERI ratio, the relative GFP, which is there, majority of the drug do not suppress GFP, but they do have the spots right here. These are the positive controls, that is the microRNA. You have some spots right here. These are the drugs which can suppress GFP selectively, which means they can induce the microRNA. So we took those candidates and then we did a dose dependency. If you increase the concentration, you see the GFP level decreases. So these are really the hits. And also we validated the microRNA expression levels. So these are the, as you see that the more the GFP goes down, you see increase in microRNA. These are the drugs which is there. I don't want to go into the details. And then we also checked the we also checked the migration of cells, cancer cells. So some of the drugs do inhibit the migration of cells when you treat them via inducing microRNA. So it's an ongoing project in my postdoctoral lab. So we are looking at the effect of these hits, these drugs on 3D models and also in vivo toxicity profiles and looking at in vivo metastasis, everything. So this is the time I came back to. I was offered the job in Central Food Technological Research Institute in CFTRI. So I came here. This is, So during this time, I realized a major thing actually, because all the drug screening what we did, the vast majority of cancer treating drugs, they have a lot of side effects. In fact, there was one paper in New England Journal of Medicine, which demonstrated, Lancet Oncology, which demonstrated that at least when you treat cancer patients with drug, at least 10% of them, they do not die because of the cancer, but they die because of the side effects of the cancer, the drugs, not because of the cancer. Which means we need to have really non-toxic drugs, not really, because majority of cancer drugs do have a lot of toxicity. Chemotherapy really dangerous for your health. I mean, we don't have an option, but it happens. So 10% of people dying just because of the drug treatment, not because of the cancer. So is there any alternative? So this is the time I moved to CFTRI. I was still really thinking about this that time. So this is the palace where we live, where we don't live. The administrative building of our, because the king of Mysore donated this building for administrative purpose. So this is CFTRI. I work in a adjunct building in CFTRI as a principal scientist. So after moving here, I had two problems. One, I have to work with the institute mandate. That is, I have to focus on food technology. So I decided, why not answer this question using food technology? And it turns out that your food has everything. In fact, some of the cancer drugs are also derived from, I think half of them or some of them are phytochemicals in pure form. And the diet what you have every day, like typical diet, the vegetables, fruits, they have loads and loads of phytochemicals, and they offer an excellent solution for any pathology, like the polyphenols, carotenoids, flavonoids, tannins, coumarins. If you look at literature, thousands and thousands of papers are there on the anti-cancer, anti-inflammatory activity of these phytochemicals. But, so we eat these vegetables, but in spite of it, every year, more than 2.25 million of new cases of cancer coming up in our country, and India is the diabetic capital. So what's the problem? Why, in spite of eating these phytochemicals, we are not able to harness their potential? That is where the key question of bioavailability of phytochemicals becomes very important, because 90% of the phytochemicals, which is not water soluble, they are not absorbed by intestine. They don't have bioavailability. Okay, that is where I was brought into nanotechnology. That's where I started working on edible nanotechnology. This is a quote from Richard Feynman saying that there's a plenty of room at the bottom. But I'm not going to work on conventional nanotechnology. I'm not working on conventional nanotechnology using gold nanoparticles, silver nanoparticles. No, they also have lots of problems. So what is my field? That's where exosomes comes into picture. I don't know how many of you are aware of exosomes. Exosomes are practically secretory vesicles from any cell type. It's just a way of throwing things out. For example, if you have a lot of dust in the house, a lot of junk in the house, you throw it in the form of dustbin. So you can consider your dustbin as an exosome, which goes out of your house. But that was the first opinion about exosomes. But it turns out exosomes are used as communication vehicles between different cell types, tissue types in your body. And they have excellent bioavailability, which means that they can really, because when you want to communicate between different cell types, you cannot just throw out naked proteins or naked DNA, anything, because naked proteins and all will get degraded in your serum or things like that, now naked DNA. So when you want to communicate, you want to send them protected. So you send them as exosomes. These are like membrane vesicles. So these are protected, which can go to the target cell and deliver a message. And exosomes are in therapeutics, and also nowadays people pack things in it of exosomes like hydrophobic drugs, cytophilic drugs, just because they increase the bioavailability of your cargo. So exosomes, or extracellular vesicles in particular, is a concerned phenomenon, which means these exosomes are made by bacteria, cancer cells, even viruses also can be considered as exosomes. So if bacteria, yeast, viruses are exosomes, or they can make exosomes, why not plant cells? Okay, so it turns out plants also have exosomes. Plants also have extracellular vesicles. This is a great plant. Actually, you inject the fixative inside this. You make a cross section, look at a microscope. You see this EV right here. EV means extracellular vesicle. You see the PVB, MV, these are MVB here. MVB is basically multivesicular body, which is a precursor for secreting exosomes out. So plants also have these exosomes, otherwise called as edible nanoparticles or edible nanovesicles. And it turns out every edible plant seems to have this extracellular vesicle. For example, you have this blueberry here, ginger here, grapefruit, kiwi fruit, orange. This is atomic force microscopy, which showing the vesicles right here. So every plant has extracellular vesicles. In the field, they are known as edible nanoparticles or edible nanovesicles. I prefer to call them edible nanoparticles at this point of time. So why is it important? It is because, and ginger nanoparticles are well characterized. Usually they have a size of about between 100 to 1000 nanometers with a peak size of 400. Depending on the plant, it varies. And they have very good zeta potential. Zeta potential is basically a parameter to measure how once you purify them, do they aggregate. The larger, the more the zeta potential, either on the minus side or plus side, they tend to have better stability. And more importantly, the phytochemicals in the plants are also in these edible nanovesicles. For example, this picture shows that gingerol and shogol, which is a bioactive constituent of ginger with a lot of anti-inflammatory activities, they are also present in this ginger-derived nanoparticles. And more importantly, let's say you give this nanoparticles, instead of giving gingerol, which will get definitely excreted, if you give them, they can get into your intestinal cells as such. That is, they have better bioavailability. They also have small RNA population. They also have a lot of things. But the take-home message is that they are orally bioavailable. So you purify these nanoparticles, you give to mice. So whatever the phytochemical which is there in these nanoparticles, they are absolutely bioavailable. And not only that, these edible nanoparticles also have microRNAs, which can target your gut microbiome in the bioavailable form. Okay? And that's the reason why several papers have come up, really high impact papers, that you purify these nanovesicles or nanoparticles. Excellent therapeutics for disease. If you orally gavage these, it can protect against alcoholic liver damage. They have a lot of anti-inflammatory activity in vivo. They can also protect mice against colonies and colon cancer. And So why is it really important? It's because compared to conventional nanoparticles, these edible nanoparticles you can get it's very cheap. You buy 1 kg of ginger, you can get 1 to 2 grams of edible nanoparticles with bioactives. And they are non-toxic. You don't have any regulatory problems with these nanoparticles because they are derived from edible fruits, bioavailable and stable during digestion. They are not affected by the gastric juice and intestinal pH, and they have excellent ADME profiles. So I decided, okay, why not focus on phytochemicals in the form of edible nanoparticles for cancer and wound healing. That's the focus I decided to work on, exploiting edible nanovesicles for targeted delivery and therapy for wound healing and cancer. So how do we do that? So basically what we do, we purify the edible nanoparticles. Depending on either you can use them as such or you can use them as a nanocarrier as well. You can, as empty shells, use them as a nanocarrier to put prohealing factors or anti-cancer compounds to target wound healing and cancer. Or you can use intrinsic factors like phytochemicals, microRNAs to target wound healing and cancer. Fine. But there was a problem. If you look at how these edible nanoparticles are purified, basically what you do, you just go make a juice of the plants. And then you, after you make the juice, you clarify the plants. We use something called differential centrifugation. You clarify the junk material, your fibers, everything by low-speed centrifugation. And then you have to purify the vesicles by something known as ultracentrifugation, which means the ultracentrifugation means it's pretty expensive equipment, probably 1 crore worth, and it can purify only 35 ml or 40 ml at a time. So this is not compatible. It's not compatible with large-scale purification, and then its cost and instrument consumables, everything is really a problem. So, and also, as I showed the main building, our instrumentation facility is located in the main building, which I showed at the palace, and our lab is somewhere else. So, you have to, sometimes if you want to do gradient centrifugation, you have to make the gradient and take it there. It's kind of a lot of logistics problems. So, I was thinking, what alternative is there? Can we purify these nanoparticles without ultracentrifugation? The idea or the clues came from, again, going back to my PhD days, purification of exosomes and viruses, because I used to work on viruses before, and polyethylene glycol is one beautiful chemical which can be used for precipitation of exosomes as well as viruses. There are several papers, several companies provide kit to purify exosomes and viruses using polyethylene glycol. So we asked the question whether can polyethylene glycol purify edible nanoparticles. So this is the hypothesis. Can we use PEG for purifying edible nanoparticles? And if yes, do they retain the same characteristics as the ENDs purified by ultracentrifugation? And the major advantage is PEG is a food additive. So you can, whatever you purify using PEG, you can eat it, no problem. So the first question is, we use this methodology. I'll not get into the details. This is ultra-purified edible nanoparticles. This is ginger edible nanoparticles. And PEG also a different concentration. You can actually purify the nanoparticles, just like ultra. We looked at the yield is also kind of similar. We can get about 2 to 4 grams of edible nanoparticles by this PEG method without involving ultracentrifugation. The size of this nanoparticles varies between 200 to 400 nanometers, irrespective of whether it's ultra or PEG. I mean, if you use a little bit more PEG amount, you purify smaller vesicles. The zeta potential, which tells you about the stability, is also kind of very similar. We also characterize them in terms of their RNA, the small RNA population. That is also very nice. Here you can see the ultra purified versus PEG purified. And just to show that these are RNA, we just treat them with RNAs, and you see there's no RNA there. So these are actually small RNA population in the nanoparticles. They also have proteins, irrespective of the ultra PEG. And the lipids also characterized by thin layer chromatography. And most importantly, these nanoparticles can self-deliver themselves inside the cells. So you just take the raw macrophages or anything, cell types, and we pre-label these nanoparticles with lipid-liking dye called Nile bread. This is the mock-treated cells. You just add this nanoparticle, within about 1 hour, you can see the cells are red because the nanoparticles gets into the cells. Actually, you can see some of the nanoparticles on top of the cells right here. This is ultra-purified nanoparticles. This is PEG-purified nanoparticles. You can see the little dots here on top of the cells. These dots are approximately about 200 to 500 nanometers. They've not gone inside. So practically what it tells you is the intracellular uptake. They can spontaneously getting into the cells, and they can deliver the bioactives into the cells without the need of any nanoemulsion or without the need of any other delivery agents. So whatever is there inside this ginger nanoparticle, like sugar and everything, sugar and gingerol, they are into the cells right now. That is kind of vindicated by the next slide, where we check whether we can once the bioactives are delivered, whether they exhibit antioxidant activities. So we check whether they can protect the cells from oxidative stress. So we take control cells. I'll not get into the details here. We take cells, treat them with hydrogen peroxide. Cells die because of the oxidative stress. But when you treat them with these nanoparticles, either PEG-derived or ultra-derived, cells are protected because the bioactives are gone inside because of the spontaneous uptake, and they're protecting the cells from oxidative stress. So this is basically the antioxidant activity in vitro in terms of total polyphenolic content and pH inhibition. So I think I have another 3 or 4 slides. So basically we have developed a very cost-effective method to purify the nanoparticles, and 10% PEG gives excellent nanoparticles without compromising on the quality and quantity. And we have extended this for turmeric as well as pomegranate. For purifying nanoparticles, it works very well. The biochemical profile of the PEG purified nanoparticles, or ENDs, edible nanoparticles, is also identical to the gold standard ultrasonification method. So now what we plan to do is, using these nanoparticles, can we modulate this microRNA FSTL1 so that we can work on both wound healing as well as cancer. In case of wound healing, we want to increase the FSTL1 content. So take skin cells and treat them with nanoparticles and see whether they can induce FSTL1, or take cancer cells and see whether they can induce the microRNA what I need. Right? So that's the way we are working on this right now. So the last part is other 3 slides left. Can we use these nanoparticles for COVID? Because COVID is kind of really picking up in the country. So as I mentioned before, these edible nanoparticles also have microRNAs. So, and they've shown that these microRNAs are bioavailable, and they can also target. This is called as cross-kingdom regulation. These microRNAs can also target at least the bacterial mRNA. That's what they've shown in a Cell House micro paper. But can we use these microRNAs to target COVID genome? So we have done some bioinformatics in silico analysis to show that these edible nanoparticles derived microRNAs can indeed target COVID transcriptome. So I think probably this might look small. The green color is basically the COVID genome, and the red color is the microRNA. And this is basically a Watson-Crick base pairing. We have actually several microRNAs in the plant edible nanoparticles, which can actually target COVID genome potentially with very high delta E values. In fact, I was looking at several drugs, phytochemical S protein interaction and all this thing. If you look at the delta E values, they will say it's 9 kilocal per mole or 10 kilocal per mole, things like that. But these are actually minus 25 kilocal per mole minimum actually. So they can really target the COVID genome with high affinity. And in fact, we have several microRNAs actually. Here the picture 8 depicts the, basically what we have done is, here is the microRNAs targeting the different COVIDs. Actually, I don't think you can see. What this figure tells you is aligning the different COVID strains, like Indian strain versus Chinese strain versus Wuhan strain versus SARS-CoV-1. Whether our microRNA, is it specific to one of the strains, or is it specific to SARS-CoV-1 not binding to SARS-CoV-2, which is a common cold virus. And we have some microRNAs which are specific to SARS-CoV-1. We have something which is really binding everywhere, like we can bind to SARS-CoV-2 also. We have some microRNAs which is kind of in between. And these microRNAs are abundantly present in some of these. This is the relative levels of these microRNAs in some of the plants here, edible nanoparticles. So, for example, tomato has about 3 or 4 microRNAs which can target COVID. The red color indicates their really high level of expression. And probably you have soybean also have about 3 microRNAs with high level expression targeting COVID. And this microRNA, GMA microRNA 16xM, is present in every plant, probably because highly conserved, which can also target COVID. So the idea here is that you purify these nanoparticles, validate the microRNAs, and they have very high levels of microRNA, and just probably develop a spray. So with that intranasal delivery, like a spray, these nanoparticles can fuse with the lung epithelial cells where actually SARS infects, the COVID, SARS-CoV-2 infects. So these microRNAs can get inside because of the bioavailability. They can go and bind to the COVID genome and prevent the replication of COVID virus. This is the kind of aspect we are currently working on. So I'll stop at that. Probably I've taken more than 50 minutes. So we'll stop here. I'll be happy to take any questions.
Any questions from the audience? Wonderful presentation, Dr. Gopinath.
Yes.
Your current work at the CFTRI, MYSORE, and your postdoctoral, 10 years of postdoctoral work in one institute. Was really hilarious to listen to various arenas of science what you have contributed, especially in the area of cancer biology. So you're really shifted from virology to cancer biology, and you really created some milestones in the area of wound healing from cancer and non-coding RNAs. And any questions to be taken from the audience? Anyone would like to ask any questions to Mr. Gopinath? Seemana?
Yes, I have two questions.
Yeah, go ahead. Go ahead, Seemana.
Okay, so the first question is, how did you find out about these plant exosomes?
That's a booming field right now. So with this work, the plant exosome actually has been demonstrated about 4 decades ago in cancer, in carrot cells actually. It's a paper in some kind of weird journal, in I think probably journal, not weird journal, sorry. It's a very old journal, 40 years ago. They showed the multivesicular body in carrot cells by electron microscopy, but nobody really focused attention because they thought it's an artifact. But in 2013, there was a Nature Communication paper where they purified these nanoparticles from grapefruits and ginger and showed that these nanoparticles can be used as nanocarriers. Like you put, like for example, you said it's difficult to transfect this U87MG cells. What you could do, you take the grapefruit-derived nanoparticles and then throw in your plasmid DNA or siRNA or anything and put it onto the cells, they'll get into them. So that was a very commendable paper I saw. Like immune cells are very difficult to transfect. They've shown that they can really get into the immune cells also. And then a lot of papers started coming in. Like at least about, I would say 30 or 40 papers have come up so far on edible nanoparticles from various plants. So that's the reason I started focusing on this. But it's a relatively young field actually.
So there is a question from G. Sirisha. Go ahead. Seemana, go ahead. Seemana, go ahead.
And the second question that I had was, like if these are already available in the plants, so why are they not available? Like you said, there is bioavailability issue, right? Like in our diet, we all consume all this, but it's not reaching us, the phytochemicals are not reaching us because of the bioavailability problem.
Okay, that I can answer by 2 ways actually. First thing is that it's about enrichment. That is, they may be there, obviously, yes, they are there, but how much ginger you eat per day? By purifying the it's same like exosomes. Exosomes are there in your body, but why do they want to use it for therapeutics? Because you're enriching them. You grow cells in large number and purify it. Then you use it as therapeutics because now it's enriched. So practically if you have a disease, I can give you nanoparticles, 1 gram of nanoparticles, which is equal to eating 1 kg of ginger. That is one way of answering it. But let me caution you, these nanoparticles are not actually purely extracellular vesicles. The reason is nanoparticles also form during the grinding. See, this, when you, basically if you take milk, when you boil it, it's kind of very separate. But let's say you put milk in a mixer grinder and just really vortex it. They're going to form these micelles. In a similar way, when you cut open the plant cells and subject them to mechanical stress, you're practically decompartmentalizing the entire cell. Everything becomes, its cell is an intact unit. But when you slice it open and just put it in a mixture and grind them like this, there also you get nanoparticles formed because of the mechanical sharing process. So purify is a mixture of both endogenous as well as which is formed during the process itself. Does that answer the question?
Yes.
So I don't really call them extracellular vesicle perfect, in a practical sense. That's why people prefer to call them edible nanoparticles, not extracellular vesicles. Yes.
That I have a question. Hello.
Yeah.
Isn't gut microflora have also ability to synthesize these edible nanoparticles, sir? Not only these plant exosomes, edible nanoparticles which are synthesized by gut flora also, bacteria, especially by bacteria.
Bacteria are known to make these small vesicles, definitely yes. But I don't know whether they are physiologically they are important or not. Maybe they may be communicating with the intestinal epithelial cells. Quite
Yes,
possible.
definitely.
I do not know actually, but I don't see any trace on that. But that's a very interesting question actually.
Yes.
Maybe possible. But in some terms of therapeutics in terms of phytochemicals, bioavailability, I feel this edible nanoparticles from plants is offering a very good, excellent alternative.
Thank you, sir.
I see a question on the, do you anticipate Dr. Garimullah Sirisha?
Yeah.
Can I answer the question?
Please, Sirisha. Yeah, please go ahead. Do you anticipate that all the nutrients in plant-based foods are delivered to exosomes or not?
With the limited repertoire which is available in terms of edible nanoparticles, probably about 20 plants edible nanoparticles have been purified, but not everything is completely characterized. My opinion on this, this completely depends on the interaction of the phytochemical, the chemical nature of the phytochemical, because it's just, you should imagine this nanoparticles as lipid layer, bilayer. Inside you might have some polar chemicals, or probably if you have hydrophobic nanoparticles, they could be inserted into the lipid membrane also, lipid bilayer membrane. So everything depends on the lipid composition of the plant and the nature of the phytochemical. So that's what I could tell you. I mean, what is the charge? What is the size of the phytochemical? So far, curcumin is there in nanoparticle, edible nanoparticles that we have demonstrated. We have it. Gingerol, sugarol is there. Even cucurbitacin is also there probably. So I can speak for few, but still a lot more characterization has to be done.
Okay, Gopi, there is one more question, Gopi. According to the literature, silver and gold nanoparticles are also got in cancer therapy, but what is it?
Can you repeat the question, please? I can't hear it properly.
Please ask. According to literature, silver and gold nanoparticles are also got success in cancer therapy, but what is the advantage with exosome?
Silver and gold nanoparticles will face
silver nanoparticles.
Yeah. First thing, toxicity. You have to do, when you, let's say you make a therapeutic formulation with silver and gold nanoparticles, you have to do toxicity studies in mice or in vivo, all these things. Second thing, regulatory problems. You have to it has to undergo a lot of regulatory issues, and some countries have also banned these, like Malaysia, I think, has banned the nanoparticles, like these kind of chemically based nanoparticles. Whereas when you use edible nanoparticles, practically it's like eating concentrated form of juice. Because we don't use any organic solvents here. It's all aqueous based. You're just making a juice and doing a centrifugation. And in fact, in the paper I showed you, the cell host microbe, they have given the ginger nanoparticles to human patients already. So you don't need any regulatory approval. What they show is that by paper is that they give it to human beings, collect the feces and show that the nanoparticles change the gut microbiome to a beneficial one. So regulatory problems is the major advantage here, and the yield and cheap, cost effective, these things. You got it?
Yeah, okay. Gopi, there is one more question.
I can hear you.
Okay, so one Sai Lakshmi has asked a question.
Okay.
mRNA level was high in pea plant. By taking that in amount per day, can we save ourselves by corona or from corona?
No, that's a problem. I said it's about enrichment actually. So as I mentioned before, these edible nanoparticles could be extracellular vesicles, probably a minor fraction, but they also form during the grinding process, during the process of purification, and it has to be enriched, just the way exosomes work, right? You have to enrich them. So you have to eat 1 gram of edible nanoparticles so that you have enriched levels of the bioactives in terms of microRNAs, which can be taken as a drug.
Okay. So any other questions? So one more question is, viruses are specific, but why COVID-19 coronavirus is transferred from bats and snakes to humans?
Actually, I don't want to answer the question because my virology is like 10 years old. So I do not know. Rather, I would say I do not know.
Okay. I think several questions are there on. So any other questions from the audience? All the people, all the participants, like, I'll send the feedback link at the end of the session. So don't panic, and you all will get So thank you, Dr. Gopinath, for spending your valuable time, and really honored to have you here today. For your major questions in science, in cancer biology, and wound healing, and cancer, and exosomes, and your current research as well. And if you would like to give any message to the student fraternity, it will be very nice to look.
Sure. Thank you so much, Dr. Brahma, for calling out to me for this presentation. Nice audience, actually. Thank you so much. Thank you, Seemana, also for a nice presentation.
Thank you, Gopi.
Yeah.
Do you like any message to the students?
Yes, actually, there's one thing I would like to say. That is because based on my experience, the reason is like my PhD was in virology, and I could have gone for a postdoc in virology again, but I sort of took a risk and took a change of field. Initially, I used to work on stem cells and differentiating them into neural precursors or MYSORE, mentorum, all this development biology. Didn't really work out that well like that particular postdoc. So we shifted to skin biology and non-coding RNAs. So that worked kind of well. So from virology to non-coding RNA, skin biology. Right now, since my institute mandates that I have to work on food edible stuff, so I work on this edible nanoparticles, where I am not a nanotechnology expert at all. But the take-home message is, whatever you are, like probably sometimes people might feel, okay, when I go for PhD, should I go for a field of my interest? Whether should I focus only on immunology? Should I focus only on this? You don't need to. You can do PhD anywhere, you can do PhD postdoc also in any field, because everything is interlinked nowadays. Everything is interlinked. Everything will have some kind of link with something else. So as long as you are good, you can actually excel in any field as long as you work hard. It's fine. Because the basic biology is same in everything. So students should not stick to any particular field. Try to explore everything. That's what I would say. Yeah.
Okay. So I have a small personal question, Gopinath. So when I
personal question?
Yeah, it's a personal question. So when I joined the lab as a postdoctoral fellow at the IISc, I had at least 5 or 6 publications. By then, you were in probably a 4th or 5th year of PhD. Yeah?
Yeah.
Yeah.
I think 4th year probably.
And I think that was your first paper on virus research.
That's right, yes.
Yeah, okay. After working for 4 or 5 years, you had one paper. And how do you feel now after having 4 or 5 Nature publications?
No, actually, let me clarify that also. It was, I have only one Nature first author and one first author in Journal of Experimental Medicine. And everything else is co-author papers. So it's not that.
Nature medicine.
Yeah, but actually, publishing high-impact journals, usually, in my opinion, there's not much difference in terms of data between 7 or 8 impact factor paper versus Nature. 2 factors are important there. First thing is your luck. Second thing, how fundamental the data, the discovery is. That's the only thing. Otherwise, in terms of data, in terms of the amount of work you put in, the hard work, is the same between 5, 6, 7 impact factors as Nature or those things. The novelty matters. Novelty and the timely, luck also favors a lot. That's what I would say actually.
Okay. Thanks, Gopi. And any questions from the audience? Any more questions to ask to Dr. Gopinath? So Gopinath will be sending our students for INSA fellowship under your able guidance. Probably should encourage some of the students working from any of our institutes. So thank you very much for spending a valuable time, especially Saturday is a day off and a holiday. And I pulled you down into the presentation. Sorry for killing your time. Yeah, and looking for some more lectures from your side to our students personally. And thank you, Gopinath. And I asked Dr. Sushila, my colleague, to deliver a formal vote of thanks. And I should also thank all my colleagues, Professor Maruti, Professor Sushila, Dr. Navina, and other colleagues from the department. They really did some untiring work to support this international online webinar. And definitely today it was a wonderful day, and large amount of audience, especially nearly around 600 to 700 audience. Lots of people are listening online in the YouTube live streaming link. And thank you very much for your patient listening. Sushila, go ahead and propose the formal word of thanks. Your voice, Sushila, your voice. Sushila, your voice. Sushila, your voice is not audible.
Okay, sir.
Yeah, now it's audible. Yeah, go ahead. Yeah, go ahead. Go ahead.
Honorable Vice Chancellor Professor K.V. Chandrasekhar Garu, respected Registrar Professor K. Krishna Reddy Garu, respected Principal Professor M. Nisha Madam, my colleagues, our most valued invited guests, researchers, academicians, dear students. It's my privilege to have been asked to propose a vote of thanks on this one-day international webinar on advances in translational cancer research and therapeutics, organized by the Department of Biosciences and Biotechnology, KRISHNA University. Foremost, I would like to thank our Honorable Vice Chancellor, sir, for gracing today's webinar and delivering the inaugural address. Thank you, sir, for your constant support and encouragement in organizing this one-day international webinar. Without your support, this webinar wouldn't have been happened like this. My sincere thanks to our beloved Registrar, sir, for his wonderful message and wholehearted wishes. I also thank our beloved Principal Madam, Professor N. Musha, for her best wishes. I, on behalf of the KRISHNA University and the entire team of Department of Biosciences and Biotechnology, extend a very hearty vote of thanks to Dr. Seema Bhattacharya Madam and Dr. M.S. Gopinath Garu, the invited speakers, for gracing their important work and sharing with us their findings and opinions today in this one-day international webinar. I hope all our participants have enjoyed the scientific sessions. I thank our head of the department, organizing secretary and convener, Dr. Veerabrahmachari sir, for his untiring efforts in making this webinar a grand success. My special thanks to all the faculty members of the department, Professor Maruti Madam, Dr. Navina Lavanya Madam, Dr. Madhuri Madam, Dr. Suresh sir, Dr. Swarupa Madam, our non-teaching staff and all our beloved students of the department for their help and support. Finally, I thank all the registered participants for their overwhelming response and valuable feedback in the chat box. Thank you one and all.
Thank you, Sushila, for your wonderful message. And shall we call off a day and let us listen to Janaganamana. If anybody has any questions, finally, to ask any questions, if anyone has missed out to ask any questions to the speakers, they are open to ask now. And you can post on the chat line. And if there are no questions, I'll go ahead and present the Janaganamana, the end song. Any message to be sent to me? I think, okay, so there are no more questions. Shall we call off a day? Dr. Gopinath, Dr. Seemana, thank you very much for your patient time. And
Thank you
very much. Yeah, let me go ahead with the Janaganamana.
Janaganu चल जल तव शुभ नाम जाने तब शुभ आशीष मांगे गाहे तब जय गाथा जन गण मंगल नायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Yeah, thank you very much for patient listening. Thank you, Dr. Gopinath. Thank you, Dr. Seemana, for sparing a valuable time, and thank you very much. Thank you very much. Yeah. Thank you. We'll sign off, and I'll send the feedback links soon. Thank you. I will send the feedback forms to all the registered emails. Okay? I'll send the link. Don't worry. Don't worry. I will send the feedback forms and certificates soon after I finish my composing and everything. Okay?
Thank you, sir.
Thank you very much. Okay. Thank you, madam. Thank you very much.